Hey there, everyone. Happy Tie-Dye Tuesday, unless you're watching this video on a different day of the week, in which case, happy day of the week to you. Today, we're going to talk about experimental design and graphing. Two parts. First one is experimental design. That's really all about how to set up a good experiment. Then part two is about graphing. So what you have to do to make sure that you can communicate your results in a good graph. This video, we're just worrying about the first thing. How do you set up a good experiment to have the best chance of getting results that actually mean something? So first is a title. When you're writing a title for an experiment in this class, we are going to use a specific format. Now, you may not use that format in other classes, and you'll see real research papers don't have titles that always fit this format, but it's a little shortcut that we are going to use just for this class. So that is the effect of blank on blank. So in the first blank, you're going to write the independent variable, whatever it is for your experiment. And then the second blank, you're going to write your dependent variable. I realize we have to talk about those words. So those are the next things we're going to discuss. And then at the end of these notes, we'll do an example that ties everything together. And I'll show you a sample experiment and a title that might go well with it. Online graphs, which you'll probably be making a whole lot more of as compared to the old school paper and pencil graphs. Sometimes they will automatically default to something like this, cuteness versus number of legs. But just a reminder for this class, we are going to have a specific format we use. So rather than saying cuteness versus number of legs, we're going to say the effect of number of legs on cuteness. So in this graph, number of legs is the independent variable and cuteness is the dependent variable. More on that in a second, but that's how we would title the graph. You may agree or disagree on the conclusions of this graph, but hopefully you agree that that's a good title for it. Okay, so next is a hypothesis. This is pretty much the same that we talked about in the last set of notes, and make sure you're using an if, then, because format. First blank, if, describe briefly what the experiment's about. Second blank after the then, that's when you predict what's going to happen. And then the third blank is because, come up with some kind of reasoning. If you are wrong about your prediction, about what's going to happen, or you're wrong about your reasoning, or you're wrong about both, that's okay. Your hypothesis may not be supported by your experiment. That's why we do the experiment, to see if you get support or not for your hypothesis. So whenever you write a hypothesis for this class, make sure you write it in an if, then, because format. Okay, on to the independent variable. Make sure you are really clear on the difference between the independent and the dependent, because that's going to come up a lot in science. Pretty much every experiment is going to have an independent and a dependent variable, and you need to know which is which when you're setting up your graph. So this is a really important concept. So independent variable, sometimes you'll see me abbreviated as IV. Sometimes you'll also hear me call it the manipulated variable. But the bottom line is this is the variable that you are changing or that you are manipulating throughout the experiment. So if you're doing different trials with a different amount of whatever, that whatever that you are changing for each trial is your independent variable. You have complete control over this variable. You're deciding how much or how little of it gets applied to each individual trial. And if you set up a good experiment, the changes in your independent variable are going to be the result, or are going to cause any changes that you see in the results. Ideally, you want to set up an experiment so changing the independent variable is the only thing that's going to affect your measurements. That's not always true, but that's always the goal. So next is the dependent variable. Dependent variable, DV, is sometimes called the responding variable. This is the one that you are measuring throughout the experiment. So because you're measuring it, you're going to set up conditions, run an experiment, see what happens, then take measurements. You do not have direct control over this variable. So if you're measuring plant growth, you can't force plants to be 7 centimeters tall, or you can't force a golf ball to roll down a ramp and come off a table at a specific distance, you kind of have to set things up and see what's going to happen. Because you are not directly controlling it, you're measuring it, we call it the dependent variable because it depends on the independent variable, or it depends on the conditions for that trial. In a good experiment, any changes in the dependent variable are going to be a result of changes that you made to the independent variable, ideally. Next are a couple things that also you need to include in every experiment. The first one is constants. So constants are other variables that could alter your results or could affect the dependent variable. And you just want to make sure you keep those the same for all trials. So if you're measuring the growth of plants, you want to anything other than your independent variable that could also affect the growth of a plant, nutrients, pot size, water, light, any of those other variables, you want to keep them the same, just so you make sure that those aren't also affecting the dependent variable. And that's in a well-designed experiment, all of those will be the same, or as many as possible. Next is the idea of a control group. Not every experiment has a control group, but a lot of experiments do. And in experiments that do have a control group, you can think of a control group as a baseline. So if you want something to compare your results to see if any changes you made had any effect compared to what the background was, you need to include a control group. Like if you want to see if a dietary supplement is causing people to gain more muscle mass, you need to include a group of people in your experiment that is not taking the supplement, because otherwise, how are you going to know if it helps gain more muscle mass if you don't have a baseline to compare it to? That's what a control group is. In experiments where you have an independent variable that's varying by different amounts, the control group doesn't contain any of the independent variable, or is just the regular conditions. Sometimes it's also called the no treatment group, but in this class, and I think most commonly, you'll see it referred to as a control group. All right, here's an example. In the next slide, I'm going to describe an experiment, and then I'm going to ask you for all the things we just talked about, but how they would be specific to this experiment. So after we go through the description, I want you to pause the video, try and write them down, and then we'll go through the answers together, or possible answers together. So here we go. A group of students is assigned a populations project in their biology class. They decide to determine the effect of colored lights on radish plants. They grow 12 radish plants in 4-inch clay pots with 25 milliliters of water daily in 100 grams of potting soil in red light, green light, blue light, and full spectrum light. Each day for 5 days, they measure the height of all the plants in each pot. If you're not sure what full spectrum light is, that is white light. So white light, like you see from most light bulbs or from the sun, the sun is actually white. It's not yellow. That's a whole nother thing we can talk about another time. I'd be happy to explain that to you. But yeah, when you're drawing with crayons, technically the sun should be white, not yellow, if you want to get all technical. But anyway, that full spectrum light, or light that appears white to our eyes, actually contains all the colors of the rainbow in it. But when they all hit our eyes at the same time, our brain kind of has a freak out, and it's like, I don't know, white light, fine. So that's what the full spectrum light is, in case you're not sure. So anyway, I want you to pause the video and see if you can come up with a title, hypothesis, IV, DV, constants, and control for this experiment. Then we'll talk about them together. See you there. All right, hopefully you actually paused it. If you didn't, it's not too late. You could do it. You'll learn a whole lot more if you actually try to answer these questions. Did I convince you? Oh, you already did it? Okay, sorry. No offense meant. Let's look at some answers. So for a title of the experiment, make sure your title is in the format, the effect of blank on blank. So the effect of colored light on plant growth, because colored light is the independent variable. That's the one that we are actually changing. And then plant growth is the dependent variable, because that's what we're measuring. Your hypothesis might be different than this, and that's okay as long as it's in the if, then, because format. So one possible hypothesis would be, if a plant is grown in colored light, then its growth will be less than in full spectrum light, because full spectrum light contains all other colors of light. Maybe that hypothesis is supported, maybe it's not, but as long as you have a hypothesis with an if, then, and a because, it's okay if you're not right when you do your experiment. Independent variable, as I explained earlier, is the color of light. If you weren't sure what unit to use for color of light, if you know a little bit of physics, you can actually measure different colors of light using their wavelength or their frequency. But if you didn't know that, that's okay. You'll learn about that in physics class someday. The dependent variable, as I said earlier, is plant height. Make sure you use units of length for that. And then also make sure you use metric units, because this is science class and we love the metric system. More on that in future weeks when we get there. Constants are anything else in the experiment that might affect the plant growth, that might affect the dependent variable. So we want to keep the pot size, the amount of water, and the mass of soil the same. Your list might even have more choices than that. And then finally, the control group is going to be the plants that we put in the full spectrum light, or the plants that we put in the normal white light, because that's going to be our baseline. Because if we only put plants in the red and the green and the blue, how do we know if they're growing more or less than normal? Or how do we know if they're not growing exactly the same? The point is, we only know if there's a positive, negative, or neutral effect if we have something to compare it to. And that's why we have the control group, which is the plants in full spectrum light. Okay, so that is an example experiment. These notes are all about how to set up an experiment to get good conclusions. In the next video, we're going to talk about how to share your results with graphing. Hope to see you there.